Okay, so in the setup video for the yeast cell experiment, one thing I didn't really show you is loading the sample onto the slide. And then in the video where I talk about focusing with your microscope, I'm actually showing you algae cells. So cells that are much larger than these cells, and they're modal, they're actually swimming around. So they're gonna look different. This is not what you're gonna see. So I need to show you in this video what it is that the yeast cells actually look like, so you know that you're looking at the correct thing. And I'm going to show you loading the sample onto the slide here. Okay, so you'll take your culture. Remember, you'll do this in the start, and then you'll do this at the other specific times that it tells you to do in the instructions. You will open up the culture, make sure you have it mixed, because I said they're not modal, they won't be swimming around, they'll fall to the bottom, so you have to make sure you mix it. You don't need very much, you only need like a drop, so you just need a tiny little bit, in here. Now you're going to take your slide. I'm going to show you two ways. I'm going to kind of hold this and bring it closer to the camera and show you what to do, and I'm going to take the camera, do it, and then I'm going to put the slide under the microscope, and we'll look at the cells that way. All right, so on the chamber, or on their slide, there are two chambers, okay? This chamber A and chamber B, this little half circle area, that's the loading zone. So that's actually where you would take your sample, hopefully this will work here, and you would be pipetting a little drop, and then you should see the liquid should get pulled underneath there. You may see it, I'm watching it move right now. It's going very slowly, but it's moving all the way across. I don't know how well you can see that, if this will focus on it or not, but I'm going to hand hold it and bring it over and do it again, okay? So that's kind of all you're going to really do. I'm going to grab the camera. Move it here. So you would see now, I'll show you this area. I did it in A. I'll do it here in B. Okay, so there it is. I put a little drop of liquid. You can kind of see here, see how that line is moving across? That's the liquid moving under the cover slip. It should become solid. I'll try to get the light adjusted right so you can see. It should become solid like that all the way across, so you don't really see any gaps or bubbles. Okay, so now what we're going to do is I'm going to take this slide and I'm going to put it onto the scope, sliding it into the base. Oops, I have this. All right, so try to line up your grid right over the little mirror. Okay, it's pretty good there. Then I'm going to put the clips. You're going to see it black out for a second because I'm going to be putting the camera clip over this. Okay. And now I'm going to be putting the microscope down. Attaching it to the scope, turning on the light for the scope. There you go. And there we have it. So now I'm going to zoom out here a little bit. I'm going to move the slide. Oh, there, I saw the grid. There we go. Okay, so this is one of the grids that you would want to be looking at. I'm going to try to focus a little here. There we go. Zoom in. Okay. Okay, so these are the yeast cells, okay, under the microscope. All right, some of them are more darker and some of them are lighter, okay. So this is what you're going to be counting. You're not going to see those little green cells like in the other video. You're going to see these cells here, And if there's a lot of them, like there are in this one, then you'll be counting the smaller squares. So you won't use the whole massive, area with all 16 big squares. You'll use, just one of these smaller squares. So there's 16 squares. You can see there's little rectangles kind of dividing them, into squares, and then super tiny ones. But you're going to be looking at these. You can see zooming in here. Those are the yeast cells, all right, that you would be looking at. Now in yours, the blue cells would be dead, and clear cells would be alive. This culture I had sitting out for a while. This is actually, it's kind of old, so I wouldn't be surprised if many of them were actually dead. But remember, you're going to be doing two different types of counts. You're going to be counting total cells, like how many there are total, and then you're going to have separate numbers of how many of the total are blue, which would be dead, and how many are alive, which would be without any color. And again, you could just take a photograph and then count it on the phone, so you don't have to be doing this live. That hopefully gives you a little bit of an orientation to what it is that you're looking for. I'm going to zoom in a little bit more here. Actually, see, it allows me to zoom in. Just going to try to refocus a tiny bit more. See, now some of these things in here are water droplets, and you can really tell. The water droplets have sort of no uniform shape to them, whereas the yeast cells are all of a very similar size and shape. Hopefully that's something that you can determine.